everybody. I'm Zoe Gage, and as I've already been introduced, I'm a screening scientist in the team at the National Phenotypic Screening Centre at the University of Dundee. I'm going to start off giving you an introduction to the phenotypic and the high content screening side of our work in partnership with the ELF, and then Jason will pick up about halfway through. So in summary, I'll start by just giving a brief introduction into high content and phenotypic screening, what it is, and the different approaches that we can take. And then I will discuss what we do and who we are at the NPSC, including some examples of assays that we've run, and go through potential assay formats and the capabilities that we have. And then Jason will pick up and talk about more detail about the collaboration with NPSC and the ELF under the SQL. So phenotypic screening in drug discovery isn't a new phenomenon. And there's an example here just of an old microscope slide of a lamb kidney biopsy. Historically, observation was the only mode of examination available. So phenomics being the study of a phenotype, and a phenotype derived from the Greek phenein, to reveal, and typos, type, and that being the composition of the observable characteristics of an organism and how these traits change in response to stimuli, being that genetic mutation, environmental conditions, or chemical agents such as chemical compounds. Screening can be defined as the systematic examination and measurement of multiple experiments. And in the context of drug discovery, those screening experiments can be biochemical, cellular, or virtual with in silico screening. So phenotypic screening then being the characteristic characterizing the disease versus the healthy phenotype. Compound screening against the phenotype to revert the disease state back to the healthy one. So the phenotypic screening approaches used in drug discovery are based on observing and defining the differences between a diseased and healthy phenotype, and utilizing that information, the assays screened against compound library with the aim of finding a molecule that, simply put, makes the diseased cell look and behave like healthy ones. This often involves multiparametric readouts that we'll discuss a bit more. So high content screening is the large-scale testing of an image-based cellular assay. That can be phenotypic, but it could also be target-based, and I'll discuss that later. But using a high throughput plate format, so the 384 well plate, we can take multiple images per well, for example, and look at different compartments in the cell. We multiplex these assays and we can quantify various outputs. So with our cell of interest, we can look over time, we can track cells, we can do live cell imaging and time-lapse imaging to get an idea of the behavior of that cell over time. We can also look at different planes once we've identified our cells of interest, and we can build up a 3D image of the volume of the cell using Z-stacks. And we can also use wavelengths and label particular regions of interest within the cell. So the nucleus, the cytoplasm, and the cytoskeleton, for example. And with that, we're analyzing the distribution, size, shape, and intensity of our compartment or wavelength of interest. High content screening, as opposed to high throughput screening, is usually a cell level analysis as opposed to well level analysis, and can incorporate parallel assays where you'll utilize the same biology but a different technology to build a better picture of the phenotype that you're interested in. So, for example, you might use the same cell line, and in one set of experiments, you'll do live cell imaging over time to track the distribution and any movements of your cells. And then you might use the same cell line to do sort of fixed fluorescent imaging to identify specific compartments within your cell. So with high content screening comes a large amount of data that's generated, and that requires high powered analytics to make sure that you can extract as much useful information out of that data as possible. So phenotypes, targets, and high content screening and how they might differ. You could take a target-centric screening approach, which focuses on a specific cellular component, compartment, process, or pathway, where an example of this might be looking at a phosphor antibody if you're reporting the activity of your kinase of interest. And on the other hand, a target-agnostic screening approach focuses on the cellular phenotype as a whole, where a well-characterized disease-related phenotype, the biology of which might be largely unknown, but you can characterize it enough to know what the behavior means. An example of this might be looking for antimicrobials, where instead of looking at a specific cellular target, you might be measuring a decrease in viral output, for example, or global viral protein expression, things like that. But most screening assays fall on a spectrum between these two extremes, and it's worth bearing in mind that a high content screen can be target-centric or agnostic. It doesn't have to be one or the other. It has to be one or the other, sorry. It doesn't have to be phenotypic. So at the NPSC, our mission is to drive and develop advanced cell-based models of human and animal disease and pathology for chemical and functional screens, where we focus on multi-component, physiologically relevant models, often utilizing primary, patient-derived, or human-induced pluripotent stem cells, for example. And we use these assays in large-scale flow and image-based assays for generating advanced imaging that and informatics for data processing, analysis, and sharing. So it's quite a busy image down here, but I'll just walk through it. So on the molecular level of screening, this is the more target-centric approaches, and biochemical assays are more high throughput and ultra high throughput assays. And if you go along the spectrum of going a bit more complex each time, you've got simplistic cell models. So this might be immortalized cell lines or cell lines derived from cancer biopsies, for example. And as we go along, we get more complex cell models and then disease models that might be co-culture systems of different cell types. And then towards the tissue models and the 3D models of a highly complex assay. So whilst this is more complicated, it's arguably more physiologically relevant, and this is the phenotypic end of screening, and this is the environment that we tend to concentrate on at the NPSC. So in order to carry out these physiologically relevant complex screens and high content screens, we need some high content readers, some powerful imaging technology, and bearing in mind the organism that we are trying to screen, we need to think about the size of the organism, what we're actually looking at, and what sort of level we need to drill down to get the information that we're looking for. So whether that be a whole organism in terms of screening parasites, worms, etc., multicellular systems of single cells, and down to the subcellular level. We have a few different readers in-house, such as that of the IXM and the InCell, which can do high throughput, high quality imaging of monolayer assays. So can the CV7000, which is our confocal microscope, and that has the added capabilities of being able to do 3D imaging through Z-stacks and also live cell imaging. And then for any assays using non-adherent suspension cells, we have a high throughput flow cytometer in the IQ, which allows us to do flow-based assays. So these imaging systems and these assays are facilitated by our modular cart-based workstations, which provide a flexible workflows on 2 robotic systems, which you can see in a graphic here. we have one larger 9-star robot and a smaller 6-star system, where the equipment you can see here is based on carts, which can be undocked and moved between the 2 robotic arms to give us a greatest level of flexibility between the assays that we can run in-house. And on these systems, we have our high throughput data acquisition readers, ranging from the confocal microscopy in the CV7000, and we go right the way down to a highly sensitive and quick multimode plate reader for a lower content screening if that's necessary. And these are fully automatable systems, so we can do fully automated cell-based screens, and this is potentiated with the use of high precision liquid handlers, such as the Echo here and the Tempest here. And we also have onboard cell storage and environmental control in an onboard incubator. And the 9-star system here has the added advantage of being completely enclosed in a BSL-2 enclosure, so we can do containment level 2 assays as well. So to give you an example of some of the assays that we've worked on at the NPSC, these have been assays that we've been working on for a number of years. So prior to our collaboration with the ELF, the complex culture systems, we've been working on a human airway model where we've used a 96 well plate format for a complex culture system at the air-liquid interface using patient-derived primary airway cells. And this culture system produces the pseudostratified epithelia of the human airway to try and recapitulate the physiological environment as closely as possible. And these growth conditions produce the differentiated different cell types that are found in the human airway, so the mucus-producing goblet cells. And we've actually produced functional beating cilia in this 96 well plate as well. Over here, we've been able to use the flow cytometer to do some exhausted T-cell assay. We've done human and mouse in the past. And then we've done some fixed monolayer assays that are more target-centric high-content screens for areas in oncology, where we've been looking at specific cellular proteins in some patient-derived glioma cell lines, for example. And then our live cell imaging capabilities are quite nicely illustrated with the male contraceptive project that we've been working on, where individual sperm cells are detected and tracked. Their movements are tracked over time, and then these movements are classified into characteristic groupings depending on their behavior. So with that, I'll hand over to Jason.
That's great. Thank you very much, Zoe. Good morning, everyone. My name is Jason Swedlow, and as Jan mentioned, I'm the academic lead for the National Phenotypic Screening Centre at the University of Dundee. Ultimately, that means I have the pleasure of working with great scientists like Zoe, who are doing or building some of the assays that Zoe just showed you. I want to start out coming back to the slide that Jan mentioned. Actually, go back, Zoe. Yeah, there you go. So you've seen this slide from Jan. I think what I would like to say to the slide from my side, so I'm just looking at the names in the call. I recognize several basic research scientists. And there really is I think in our community, there is a huge desire to take the systems and the knowledge and the capabilities that we're developing in the basic research side and move them towards, try to take those capabilities and move them towards the development of new drugs. But the fact is that that's actually, as we all know, a quite significant challenge. And this slide highlights the number of years that it can take to get through all of the necessary steps. And in particular, the first couple of steps, just getting good hits to take into a drug development program is a significant barrier. And so ELF, and in particular, as we're talking about today, the high content screening side of it, is really an attempt to lower the activation barriers for those early steps and make it possible for academic basic research scientists, SMEs, to really drive forward their discovery level research into drug development. That's really the overall mission of the European Lead Factory. So that I think that's a really important opportunity for all of us. So in the next slide, so if you wouldn't mind, there you go. So Jan mentioned that the ELF has put together over the last several years a pretty substantial library. And I have to say this slide emphasizes 2 points. First of all, the size of that library, and you can see the number of compounds that are available. But most importantly, at least in my opinion, is the fact that in the construction of those libraries, you're accessing the knowledge of chemical space and expertise from a wide range of pharmaceutical companies, and then bringing that library together into a unique resource. At least Jan may correct me, but to my knowledge, the only place that this library is available is through the ELF. And so that, as I said, basic research can access that chemical knowledge and use that library to define hits to take forward for drug discovery. Now, you see some pretty impressive numbers in terms of the size of the library there, and in other projects, those larger libraries are used for high throughput screening. In the context of high content screening, as always said, in many cases using advanced biological models, we're using a smaller subset. So the ELF has worked with its partners to define a 50,000 compound subset that covers the chemical space, representation, et cetera. And so that is the library that we're screening projects. We're using to screen projects within our collaboration with the European Lead Factory. So next slide, Zoe, please. So, what are we actually doing? And in particular, what's in it for you? So first of all, I want to maybe start at the bottom right of the slide. I think the most important take home of this presentation is if you're interested in any of the programs we're talking about here, there are application forms that really go through an awful lot of details. And just even looking at those application forms, I think would be useful to get an idea of what we're looking for in the screens that we take forward. What do you get out of this? Well, first of all, we have funding for a lot of the costs for running these screens. So as I mentioned, the ELF has constructed the screening library and making it available to scientists who are using the screens. And so normally, if you came, for example, to NPSC or another screening center and said, well, I want to run a drug compound screen, we would charge you for the library. I have no idea how to price the value of this library because it's so special, but that's covered. Staff time is covered, so people like real experts like Zoe and her colleagues, their time is covered, and we have reasonable costs for consumables, et cetera, that are covered within the project. So you saw the facilities that we have that we can make available within NPSC that Zoe showed you. What we produce for you using the assays is that you've developed our initial triage of primary hits, and what we're going for, the ultimate output of this project would be what we call a qualified hit list of up to 50 compounds that have come out of that 50,000 compound library. And that qualified hit list should be a really great starting point for further drug development. In the process, the necessary resynthesis, retesting of the most promising hits is all included, and a report of all the results is handed to you. Jan can tell you more about the details of this, but you, not us, but you have access rights for 3 years to exploit the results in concert with the partners that are the source of the compounds, and you really decide how to progress all of this. And I think again, this final point I think is a really interesting one. I think the opportunity to start to be partnering with quite serious and quite accomplished drug development efforts pharmaceutical companies and the like is a really critical part of this. And so just, the connection and the networking and the capability to bring your great science into a discovery path that generates this qualified hit list and then starts the discussions with pharmaceutical companies is a really great feature of this program. I'll go back up to the first point. So screen against our compound library at no upfront cost. We should qualify that. So, there are, you can imagine constructs of screens which would be very expensive. And as we'll talk about in a second, I think in the, if the price of the, for example, the reagents or for example, the cell lines, et cetera, is too high, then we can work together to define ways to drop the cost so that we can actually run that 50,000 compound screen at scale. So Zoe, if you would, to the next slide, please. Okay, so this it highlights the workflow that we'll go through together if you submit an application to this program. So as I mentioned, the application forms are all on the ELF website, and you can find them there. We found that the application form, defines an awful lot of what we're looking for. We'll talk about that more. There's an expert review process that the ELF has commissioned that then reviews that application, and obviously that application is then either approved or revisions are requested or ultimately rejected. One thing I want to say about that, I sit on that review committee, and just speaking quite personally, it's been a real eye-opener because it's the first review committee I've been on where there are academic biologists, scientists, so there'll be cell biologists, but also pharmaceutical scientists, chemists, and so it's a really eclectic mix of reviewers who are going through the applications. And so I think it's a real opportunity to have your ideas tested against a broad set of expert reviewers and to get feedback. In many cases, we do ask for revisions, and then we can look for resubmissions. Ultimately, if the project is approved, then there is you'll work with Jan, Vera, and the team at Literature to define the project definition. We at NPSC will contribute into that discussion from a technical point of view, and then we move into an assay development step, and I'll talk a little bit about that more in the next slide. But ultimately, we're taking your technology, your assay, and putting it into a scalable format is probably the shortest way to say that. And we do that with what we call an annotated set. So these are public compounds, a relatively small set, 3,000 compounds. The key there is to generate a really robust and a scalable assay. So if we, and if that succeeds, it's not guaranteed that will succeed, but if it succeeds, we'll then take the assay forward into the full production screening. Now, in the worst case scenario, let's pretend for some reason we simply cannot scale up that assay. What you will receive is a report of your assay run against these public compounds. So public compounds are known compounds, and in many cases, at least, the targets of those compounds are known. So hopefully that's useful information. So that would be the worst possible outcome of this process, which I can tell you from a basic cell biologist point of view, I think that would be pretty great. Assuming that we are able to scale the assay, and we have had a lot of success there, we then take that forward, and that feasible at scale sort of question is really about the assay metrics. We'll talk about that more in a second. We then move into that production assay with the 50,000 compound library that I mentioned, dose response curves, resynthesis tests, et cetera, ultimately leading to the production of what we in the project call the QHL, or the qualified hit list. I know that's an awful lot of words there, so if anybody wants to ask any questions about that, happy to explain that more. So if you would, to the next slide, please. So what can we do? Zoe showed you a lot of the technologies that we have in-house, but just just to give you an idea of what we're looking for in terms of assays and the technologies that we can meet. We're really looking for assays that can be run in at least a 384 well format. We have lots of different imaging modalities, and many of them mentioned there. And I do want to highlight that we can work with either attached or suspension cells using the flow cytometry capability, the robotic flow cytometry capability that we have. Lots of different readouts, a full data management and data processing. We have a reasonable computational capability at NPSC as well to do the necessary analysis. And I direct you to the URL at the bottom of the slide there for more details on all of these. Zoe, next slide, please. Okay, so what are we looking for in that evaluation process? And so I suspect a lot of this won't be surprising, but maybe there's a few details here that you want to keep in mind. So obviously, and keep in mind that ultimately we're developing compounds, we're hopefully identifying compounds, excuse me, that can provide a partnership between yourself and one of the pharma partners in the project. So is the target or the approach novel, and is there a clear need? So is there a need for the pathway that you're interested in, for example, as a drug target? And the second, and that sort of leads into the second point as well. Is there really high scientific quality here? Is this excellent science that's underpinning the idea of the assay? Can we actually scale the assay and make a true high content screening assay and do it at automated scale? That's absolutely necessary to run the size of libraries we're talking about. And just to flesh that out a little bit, when we run the assay, can we get robust quality control metrics? There's various words there that if you're in the assay world will make sense. If signal over background, Z prime, CV, etc., aren't immediately familiar, I again direct you to the website, which has nice definitions of all of those, and so hopefully that's helpful. And if you look at the application, we'll ask you explicitly for those parameters for you to supply example parameters for the assays at the scale you're able to achieve currently. So what that comes down to is scientific quality. If there are targets involved, do those targets make sense from a chemical point of view? Is it tractable to imagine that we can develop chemistry targeting this process? We obviously want innovative assays. A differential compared to existing treatments or existing pathways that are already in the market, and ultimately, whether these can, whether if we were able to develop really great hits, whether they could be exploited. And again, this is all on the website. And so, next slide, Zoe. So I hope that summary is helpful. I just want to highlight one of the greatest things about this project from my standpoint, and just an enormously exciting collaboration across many different types of entities. This is the first time that in my career that I've been I've had such a interesting collaboration with so many pharmaceutical companies, CROs, and all trying I should say, and also academic scientists, all trying to build this capability that we're telling you about today. I should also just acknowledge the efforts of Jan, Vera, and the team at Ligashare, who are behind the scenes making all of this happen. And it's possible from this slide, if you can imagine this many different organizations connecting and working together, is a real challenge, and they make it all happen and actually make it look easy. So with that, I think the next slide is just a thank you. So yeah, thank you for watching. I hope that was helpful. I hope what Zoe and I presented to you today was useful. I've seen lots of questions popping up, but I haven't been able to read them, but I'm happy to turn to those now, or I think Vera wants to come in and mention a few things, then maybe we'll come back to questions. Thanks a lot.